This is Unit 3, Topic 6 and 7, Cellular Respiration. Let's get started. So before we start, let's take a minute to review your notes or the OpenStax textbook online for the structures of mitochondria. You can see we have ATP synthase particles, which we're familiar with already. The matrix is what we call the inside portion. You have the intermembrane space, or I should say the intermembrane space. That's the space in between the inner and outer membranes. The fold parts here we call cristae, ribosomes, that's just your general organelle, granules, DNA, you can see the DNA circular. Mitochondria has its own DNA, it's circular. Then you have the inner and outer membranes. So we are looking at the mitochondria for this lesson. We are talking cellular respiration. So cells harvest chemical energy stored in organic molecules and we use it to generate ATP. In plants, these organic molecules come from photosynthesis. Remember, the final product of photosynthesis is a molecule of glucose after it goes through, the Calvin cycle. So organic molecules plus oxygen usually came from the environment equals CO2 plus water and energy. Starch is the major source of fuel for animals. This breaks down into glucose. So animals like you and I, these heterotrophs, we use oxygen and organic compounds to go through this process of cellular respiration to get energy, to do things, to grow, to divide, to just exist and live. Cool. I should mention too, this is a common misconception. If you're taking a look down over here at the cycle, both heterotrophs, we get organic compounds from the environment, oxygen from the environment, and go through this process. But plants, autotrophs, they also go through this process. This is a process that happens simultaneously with photosynthesis, but in conjunction with. So common misconception, plant cells that photosynthesize, autotrophs, still go through cellular respiration. So here's the catabolic breakdown of glucose. So you can see here C6H12O6, that's a molecule of glucose. You add 6 oxygen molecules to that, and you get 6 carbon dioxide molecules, 6 water molecules, ATP, and heat. Remember, Leo goes Ger, so oxidized reactions are losing an electron, and reductions are gaining. So for example, the oxidation of glucose transfers that electron to a lower energy state. So this releases energy to be used in ATP synthesis. So you can see oxidation, reduction reactions tend to couple with each other. So where glucose is losing a molecule, O2 is gaining a molecule. So pretty cool. Explain the difference between catabolism and anabolism as it applies to the overall equation of cellular respiration. Remember, my cats break things. Catabolism is the breaking of a molecule. Anabolism is the building of one. So let's go back to this equation here. You can see that glucose here is being broken down and O2 is being built up. Cool. Path of electrons and energy harvest. So during cellular respiration, most electrons will flow in this downhill exergonic path. Remember, exergonic releases energy. X meaning out. So you start with glucose, NADH, electron transport chain. Finally, you get oxygen. This is just like a super basic general overview of what we're going to get into. So let's talk about energy harvest. Glucose is broken down in steps to harvest energy. Electrons are taken from glucose at different steps. When electrons are taken, they travel with a proton. Remember, when we talk about passing electrons, we're really talking about a molecule of hydrogen. This is how we pass electrons in biology. Dehydrogenases with the ASE, you can see it's an enzyme. This enzyme takes 2 electrons and 2 protons from glucose. Whenever something takes an electron, like an enzyme over here, we call it an oxidizing agent. So dehydrogenases are oxidizing agents for glucose, and it transfers these electrons and 1 proton to a coenzyme. Remember, a coenzyme is a molecule that helps another enzyme, and it is proteins. It is an organic compound. So the coenzyme to the coenzyme NAD plus, and it reduces this. Remember, reduction is gaining an electron, so you can see this is a positive molecule. When we put an electron on there, it becomes NADH. This is a molecule that stores energy. As long as that hydrogen's bonded onto there, we have a little bit of energy storage. If we need that energy, we can release it. Other proton is released into the surrounding solution as that hydrogen right there. So you can see how that hydrogen gradient can start to build. NADH then carries that electron to the electron transport chain, which we're already kind of familiar with. So a pair of molecules that are oxidizing reagents, or oxidizing agents, I should say, things like dehydrogenases that help carry electrons. So if you guessed FAD and NAD+, you're good. So how do we harvest this energy? So we just kind of started the basics of where this energy comes from and what carries it. The energy harvest starts at the electron transport chain. You guys are familiar with this. It is a similar mechanism to what we saw in photosynthesis. It's a sequence of membrane proteins, and they shuttle electrons down a series of redox reactions. Remember, redox is reduction and oxidizing reactions. One molecule will lose an electron, another will gain. It releases energy used to make ATP. Similar to photosynthesis, the electron transport chain transfers electrons to oxygen, the final electron acceptor, to make water. Oxygen being the final electron acceptor is an incredibly important piece of knowledge to know for our exam. And we'll go over it in more detail. You're going to see an image of it, so it might make a little more sense. But in cellular respiration, oxygen is the final electron acceptor. It's just a molecule that grabs that electron in the end. This releases energy. Yep, releases energy. So let's get into the whole thing. Here's stages of cellular respiration. There are some stages that we need to know. There are some that take place in the cytosol, some that take place in the mitochondria. 3 stages in particular: glycolysis, right over here, that takes place in the cytosol outside of the mitochondria. Pyruvate oxidation and the citric acid cycle, they couple together, okay? And those happen in the mitochondria. And then finally, oxidative phosphorylation. That is a combination of the electron transport chain and chemiosmosis. So some of these words might sound confusing, totally fine. Oxidation is oxygen, citric acid cycle, oxidative phosphorylation. We know that phosphorylation is the adding on and the removing of phosphate groups, electron transport chain. These are words that we can kind of break down and become familiar with. So let's start with glycolysis. This is the starting point of all cellular respiration. It occurs in the cytosol. So if you can imagine, this is like kind of zoomed in on the mitochondria organelle. There's another big cell around it. And this process begins in the cytosol. This is a process where glucose molecules, which are generally 6 carbon molecules, are split into 2 molecules called pyruvate. All you need to know is 6 carbon splits into 3 carbon. That 3 carbon we call pyruvate. Here's the whole process. It looks like a lot, and it is a lot. The fun fact is, you don't need to know every single step. Don't need to know that until college. So all you need to know over here, let me get my little tool, is that we start with a molecule of glucose up here, and it has 1, 2, 3, 4, 5, 6 carbons. See in that circle right there? All right. And something happens to it, and something happens to it, something happens to it, and all of a sudden we have 1, 2 different molecules. So we start with 6, and we split it into 3. It goes through a whole other process over here to be modified and do other things, but you can see at the end product here is a pyruvate molecule right up here, and another pyruvate molecule over here. So another thing to notice here, other than the 6 carbon to the 3 carbon, is that at each stage we need energy, right? In order to split a molecule, we need energy. So ATP, is going to be converted to ADP in every single step, okay? In the end over here, and we'll get into it because there's the, we need to use energy for this step, but in the end over here, we start gaining energy, okay? That's sort of your redox reactions. So things to know, 6 goes to 3, 6 carbons go to 3 carbons. That 3 carbon molecule is called a pyruvate. This is essentially what you need to know. Here's the summarized pathway. You need to know the inputs. As I mentioned, it's glucose, 6 carbons. You need to know the outputs, which again is pyruvate. Another output, so another product here, it's 2 molecules of water, 2 molecules of ATP, 2 molecules of NADH, and 2 molecules of hydrogen. Okay, so glucose comes in, ATP is formed, NADH is formed, H2O is formed, and pyruvate is formed. 1 glucose into all these. There's 2 stages, as I was mentioning in the earlier model. There's the energy investment state. So the cell has to use ATP to phosphorylate these compounds of glucose. And then there's the energy payoff stage, where energy is produced by substrate level phosphorylation. So let's go back, let's look at this, right? So this is your energy investment stage where we need energy. We start with ATP and we end with ADP that releases energy. And then here is our energy payoff stage where we start with ADP and hydrogen and we end up with ATP. Awesome, good stuff. There we go. Energy investment and energy payoff. The net energy yield per 1 glucose molecule, so again, we have to like pay into it and we only get a little back, is 2 ATP molecules, 2 NADH molecules, right here. Okay, so 1 glucose equals 2 ATP, 2 NADH. That's the big, that's another big important one. So summary, energy investment, 2 ATP molecules going into 2 ADP molecules. Here's our payout right there. Here's our net. So at the end of this whole process, this is our products. 1 molecule of glucose equals this stuff during glycolysis. As you can see, the arrows are now pointing, so these products are now going to assist us in the next step. Where does glycolysis occur? The astute scholar would know it, and the answer is the cytosol. What is produced through glycolysis? You may need to flip back in your notes for this. We got pyruvate. What is the net production of ATP? Again, you may need to flip back. 2 ATP. Fun fact, 2 NADH as well. Great. So let's take those products, go into pyruvate oxidation. So if oxygen is present, the pyruvate enters the mitochondria. Pyruvate is then oxidized into acetyl-CoA. This is a big overview. Acetyl-CoA is used to make citrate in the citric acid cycle. 2 CO2 and 2 NADH are then produced. Here's the overall. Okay, so you have your input of pyruvate. Acetyl-CoA is a coenzyme that'll help with us. That'll help us with this reaction. Oh, I'm sorry, CoA is over here. Acetyl-CoA is our product. So start over. Pyruvate is our input. NAD plus is over here. This gets reduced, plus CO2. CO2 is our product. Acetyl-CoA is also our product. So this is like your big picture overview. Remember, this pyruvate comes from the glycolysis process. Citric acid cycle is also part of this too. Remember, we lump those two together, the pyruvate oxidation, because pyruvate oxidation is essentially just this one step, right? Where pyruvate, we make some energy. helps out, product, product. Citric acid cycle, we're going to lump this together. We also call it the Krebs cycle. You can hear it as the Krebs cycle too. This occurs in the mitochondrial matrix. So remember, we have the outer membrane, we have the inner membrane, outer membrane, and it occurs in the matrix. Matrix is the stuff on the inside. It turns the acetyl-CoA into citrate. So remember, we started with glucose. Glucose passed the pyruvate over to here. Pyruvate was oxidized, turns into acetyl-CoA. Acetyl-CoA is now passed over here. And we turn that into citrate. When we do that, it releases CO2. We make ATP, and electrons are transferred from these other two high energy carrying molecules, NADH and FADH2. So ATP is, I should also mention this too, ATP is the major energy carrying molecule in the body. NADH and FADH2 are similar molecules. They function similarly to ATP. They just don't give off as much energy. They release this hydrogen here, which hydrogen bonds don't hold as much energy as phosphate bonds. So here's the whole process. I know, it sucks. It's confusing. Things to note up here. First of all, pyruvate's right up here. CoA is over here. Acetyl-CoA is our product. Then the citric acid, so this up here, this is sort of your pyruvate oxidation. Okay. Once we get the product of your pyruvate oxidation, it goes into this cycle over here to make, it starts with citrate, and a series of reactions occurs, and it goes around here in a cycle. That's pretty much all you need to know for the purposes of the AP exam. Some other things to note over here, you can see there's some formations of NADH, there's some high energy molecules. GTP is another sort of helper in this process. Yeah, that's about it. Okay, so it looks confusing. We're going to go over it in a high-level overview. Here we go. This is a little bit easier, right? Summarized pathway. You really need to just know the summarized pathway. So acetyl-CoA, right up here. This is the product of pyruvate oxidation. Goes around in a cycle. So you got your inputs, your acetyl-CoA molecule, and what we get out of this cycle is 2 molecules of ATP, 6 molecules of NADH, 4 molecules of CO2, and 2 molecules of FADH2. So, quick check. Where does the citric acid cycle occur? It's in the matrix, like Neo, except it's the mitochondrial matrix. What is the net production of ATP in the citric acid cycle? You can go ahead, flip back. 2 ATP. Okay, those are the only 2 quick checks. All right, last part of this process. The electron transport chain and chemiosmosis. We call this oxidative phosphorylation. It consists of, as I just said, the electron transport chain and chemiosmosis. Here is the whole electron transport chain in the mitochondria as it applies to cellular respiration. You may notice a few things. It looks similar to photosynthesis. You have the inner mitochondrial membrane as opposed to the thylakoid membrane, but you still have these intermembrane proteins, one right here, one right here. 1, 2, 3, and 4 is what we call it. I know, so much easier. You still have ATP synthase over here. It's the same molecule that we saw in photosynthesis, the same protein. It does the same thing, except the hydrogen molecules moving out here, they move into here. But it does the same thing. It creates this ATP. All right, here's in your mitochondrial matrix, it picks up 2 hydrogen molecules, 2 electrons, half an oxygen molecule, and it makes water. Process goes through twice to make a single molecule. So this is kind of the high-level overview. Let's get into it. So it's located in the inner membrane of the mitochondria. It's a collection of proteins, just like we saw in photosynthesis. As the same thing in photosynthesis, there's an electrons that are built up over here, and as they fall, proteins alternate. So over here, electrons, 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 they go up into here. As they fall, they alternate between reduced and oxidized. So again, this is another redox reaction. where one molecule accepts an electron and another donates. You can see this right here, an acception and donate. What is the role of electrons in the electron transfer chain? To dephosphorylize ATP, to power active transport pumps, to reduce heme in complex 3, or to oxidize oxygen. If you said to power active transport pumps, there you go. These are the powered of the transport pumps. Okay, same over here. So one goes one direction, one goes the other. Follow the arrows. All right, the cristae, the folds, the inner membrane folds of the mitochondria increase the surface area for reactions to occur. We saw that this is really important and really good. The more ATP, the more energy. It does not produce ATP directly. ATP is produced by going through this process. Instead, it helps maintain the release of energy by creating several small steps for the electrons to fall through. So again, there's a buildup of electrons on this side. They fall up, and then they fall back down. When they fall down, they make that ATP. The final electron acceptor, we mentioned this earlier in our high-level overview, but it is incredibly important, is oxygen. This is the reason we breathe, essentially. So the process occurs when each oxygen pairs with 2 hydrogens and 2 electrons, and we form water. So it occurs in the mitochondrial matrix on the inside. You can see picks up 2 free hydrogens, 2 electrons that already exist in the electron transport chain, half a molecule of oxygen. The process goes through twice, and we end up with H2O. One major function of the electron transport chain is to create a proton gradient across the membrane. As we know, proton gradients are incredibly important. These proton gradients occur as proteins shuttle these. Sorry, I need to get up and close the door. As they shuttle electrons along the electron transport chain, they also pump hydrogen into the intermembrane space. They use this exergonic flow of electrons. Remember, exergonic flow is like releasing the energy. X means exit or out, sort of like exocytosis or exergonic, as we see here. That exergonic flow of electrons moves from NADH to FADH2. This gradient will power the process of chemiosmosis. Okay, so remember I had said that the electron transport chain and chemiosmosis are paired together. Chemiosmosis uses the hydrogen ions to power cellular work. That's things like metabolism, growth and development, mitosis, anything the cell has to do. Cells work too, 9 to 5. No, I'm just kidding. They're 24/7. All right, cool. So you can see over here, they are pumping hydrogen out. These are the proteins in the inner mitochondrial membrane, pumping proteins out from the matrix right over here. Boom. So this is sort of the process of chemiosmosis. It's that pumping it out. What would be the outcome if hydrogen ions were able to diffuse through the mitochondrial membrane into the mitochondria without the need for intermembrane proteins? We encountered a question like this recently with photosynthesis, where like if the membrane proteins were just open and passive transport was occurring, what would be the outcome? ATP wouldn't be produced. Done, right? If this process isn't occurring, if these hydrogen ions can just move freely, then they're not going to be pushed through here, and this process won't occur, right? Without that process, we don't get ATP. We don't have energy. Amazing. Chemiosmosis is this, right? So electron transport chain is passing these electrons, pumping these ions out. These electrons out, and them falling down through this, falling down their gradient, as we say, from high to low concentration, producing that ATP. So ATP synthase, we've seen this enzyme before in photosynthesis. It's the enzyme that makes ATP from ADP and inorganic phosphate, and it uses the energy from that hydrogen gradient across that membrane with high concentration over here and low concentration over there to drive sort of this chemical process. So hydrogen ions flow down their gradient, as I mentioned, through ATP synthase. It acts like a rotor. Oh, yeah, I like that analogy. It acts like a rotor. So when hydrogen binds, the rotor spins. This activates a catalytic site that turns ADP into ATP. So essentially what they're saying is that, so as things move down like this, it's sort of, this process kind of goes one to the next, one to the next, one to the next. So like one goes down and the cycle goes. One goes down and the cycle goes. Pretty cool stuff. So this is a huge process. This is your biggest energy producer in this entire concept of cellular respiration. It produces a ton of ATP. So 26 to 28 ATP molecules per glucose. All up until this point, we've been working with like 2 or 4. This is a huge process. It produces a lot of energy. Cellular respiration, what is moved? What does this occur? Hydrogen ions across the mitochondrial membrane. These check for understandings are old. They're still useful. Okay, quick check. How is a proton gradient formed across the inner mitochondrial membrane? You guys know this. Exergonic flow of electrons, NADH to FADH2, powers the complexes in the electron transport chain to pump the hydrogen into the intermembrane space. So energy is released, electrons are pumped in. This is an exergonic process because remember, we are pumping ions against their gradient. They don't want to go into that space. There's too many of them in that space. So we have to have an energy output in order to get them there. This is an exergonic flow of electrons, right? And as the hydrogen pumps go into the intermembrane space, there we go. Similar process that we saw in photosynthesis. It's just a different organelle that it's happening in. Final electron acceptor of the electron transport chain. We're going to like chant this in class. The final electron acceptor in the electron transport chain is, oh my goodness, oxygen. It's got to be oxygen. How does ATP synthase obtain energy to convert ADP, an inorganic phosphate, to ATP? Well, you would know that's that proton gradient, that hydrogen ions that are falling through there, acts like a rotor, goes ADP, ATP, ADP, ATP. Cool. Here's the whole process in all of its glory. If you can remember this, I would add in another column with like, where does it take place in the cell? Like, is it in the cytosol? Is it in the inner mitochondrial matrix? Is it in the membrane, whatever? If you can remember this right here, you are golden, okay? You need to know the inputs and you need to know the outputs. So just taking a look from top to bottom here, 1 glucose in the end produces 30 to 32 molecules of ATP. Goes through this whole process to do that. And that is inputs, outputs, that is energy investment, energy requirement stage, like it's all, or energy output, it's all right there, 30 to 32. What happens in molecules, I'm sorry, not molecules, organisms, with no oxygen? They go through this process of respiration. So remember, in cellular respiration, oxygen's the final electron acceptor. It drives electrons down the electron transport chain, it accepts that, and it creates water, correct. How do organisms produce ATP in the absence of oxygen? And there are plenty of organisms that can exist in the absence of oxygen. Fermentation, we'll get into it, but aerobic respiration is what we call, or anaerobic respiration is what we call it. A in front of anything or AN in front of anything means not. Aerobic, think about aerobics, like going to do like aerobics, as like a workout, needs oxygen. And then fermentation. So anaerobic respiration generates ATP using the electron transport chain with no oxygen. Pretty cool stuff. It takes place in prokaryotic organisms that live in environments with no oxygen. That could be anything from a prokaryotic organism at the bottom of the ocean to, the yeast that's hanging out inside of a, kombucha. A final electron acceptor in anaerobic respiration are generally sulfates and nitrates instead of being oxygens. Fermentation is a similar process. Again, it generates ATP without an electron transport chain at all. They just use it as an extension of glycolysis. It still recycles NAD+, occurs in the cytosol, so no oxygen present here. Two types. There's alcohol fermentation, and there's lactic acid fermentation. Lactic acid fermentation occurs actually in your muscles. We'll talk about that if I don't do this here. Type of cellular respiration is represented here. Anaerobic, so it's not going to be aerobic because there's no oxygen. Anaerobic, NADH donates. Electron receptors, inorganic, it's this one. Oh no, that's an organic electron acceptor. There you go. Anaerobic respiration, because NADH donates its electron to a methane molecule, which is CH4. Okay, alcohol fermentation. Here's the whole process. So pyruvate, so we still have glycolysis that occurs from glucose. So glucose is transferred into two molecules of pyruvate through glycolysis. That pyruvate is turned into ethanol. There's a intermediary step over here that it turns into acetaldehyde. It creates carbon dioxide molecules, and we use NADH as our high energy electron carrier. Bacteria, yeast do this. Yeast go through alcoholic fermentation. This is your kombucha, right? If anyone likes kombucha, this is your probiotics. Cool. Yep, that's all you need to know. Lactic acid fermentation is also really interesting. Pyruvates reduce directly by NADH to form lactate. So lactate is an acid. Did we talk about this in here? Okay, yeah, we do. So glucose, again, goes through glycolysis. Two pyruvate molecules are our product, and it goes through this simple process of reduction from NADH to NAD, from NAD plus to NADH, and we produce two lactate molecules. An example of this is your muscle cells. When muscles run out of oxygen, they go through this lactic acid fermentation to produce ATP. It causes a burning sensation when you are performing strenuous exercise. Also, interestingly enough, so lactic acid is exactly that. It's an acid. And if you are the type of person who not the type of person, but if you've ever held your breath for a long time, you can kind of feel like your muscles almost like burning, right? That burning sensation is literally your blood turning acidic because it goes through this lactic acid fermentation. It happens pretty quickly because you don't have any oxygen when your muscles run out of oxygen. So when you're exercising and you feel that that burning sensation in your muscles, that is because your muscles aren't getting enough oxygen. Cramps are a part of this too, not like menstrual cramps, but like cramps when you're working out, right? This is a part of this too. If you feel that burning sensation, stop, take a couple really deep breaths, get some oxygen into there. Your cells will start going back into cellular respiration instead of continuing with lactic acid fermentation. It's so cool. I mean, it hurts, but it's cool when AP biology can relate to you. Cool stuff. Muscles will produce lactate, which goes into the blood. We were just kind of talking about this. It's broken down back into glucose with the liver. When lactate's in the blood, it lowers the pH. So again, you're literally turning your blood into acid. If lactate builds up and is unable to be broken down, we call this lactic acidosis. Acidosis, you'll learn this if you take biomed with me next year. Acidosis is excessively low blood pH. Ketoacidosis is another sort of disorder that can happen over here. Ketoacidosis comes from low glucose. It happens a lot in patients with untreated diabetes. But yeah, pretty fascinating stuff. Okay, fermentation methods that can occur in animal skeletal muscles. Lactic acid fermentation. Okay, here is putting it all together. This is sort of like a flow chart. So everything starts with glycolysis in the cytosol. So we eat food as heterotrophs. Autotrophs just went through photosynthesis. So we have a molecule of glucose that's going to enter glycolysis in the cytosol. If there's oxygen present, so if we're breathing, if we are, we got oxygen, we go through this process. Pyruvate oxidation, where that glucose molecule is broken down into two pyruvate molecules, the citric acid cycle. and those products then go into the electron transport chain and chemiosmosis. This process over here produces 30 to 32 ATP molecules. Great. If you are a heterotroph bacteria, you just ate some other organism that was really tasty and you got a glucose molecule. If you are like a cyanobacteria that photosynthesizes, yay, you just got a glucose molecule from photosynthesis. No oxygen is present. It's going to go through fermentation, anaerobic respiration. Two different types: lactic acid formation, which produces 2 ATP molecules, and alcohol fermentation, again, 2 ATP molecules. So you can see the big difference between oxygen and no oxygen as far as ATP output goes. So you can kind of see why, evolutionarily speaking, you're going to want to be up here with the oxygen, right? This includes like aquatic organisms as well, because there's still oxygen dissolved in the water. We're talking like bacteria and protists and that sort of thing that don't have any oxygen. Tardigrades, right, that are living in the middle of space. Cool. So you're going to want oxygen. It's important. Okay. Yeah, okay, yep. I want you to do this practice FRQ on your own. We'll talk about it in class. This one too. That's it. Cool. Thanks, guys. Remember to pass your notes in, and I'll see you in class.